Hi everybody, DrBiology here, and this is the part 2 of biological molecules, and I'm going to be looking at carbohydrates today. Okay, so if you saw part 1, monomers and polymers, here's a bit of short-term retrieval. Okay, so I'd like you to look at the questions, pause and answer. Okay, so what is a polymer? Well, it's many monomers joined together. What reaction joins two monomers together? That will be condensation. Which reaction breaks down polymers? It's hydrolysis. And what type of chemical bond is formed between monomers? And that is a covalent bond. And it obviously differs with the type of biological molecule. Okay, so with carbohydrates, we need to first talk about monosaccharides. So these are monomers from which large carbohydrates are made. There are three monosaccharides you need to know about, which is glucose, galactose, and fructose. As you can see, they have the same molecular formula, which is C6H12O6. These are called isomers as they have the same chemical formula, but the atoms in the molecule are attached differently. Obviously, glucose exists in different forms called structural isomers or isomers. So two common isomers, though, for glucose are alpha glucose and beta glucose. And you can see the big difference is the H and OH is swapped over for alpha glucose and beta glucose. The reasons for that will become clear when I talk about different examples of polysaccharides. Two monosaccharides combine together to form a disaccharide. So as you can see in this picture, the bond that forms it is called a glycosidic bond. And the reaction is called a condensation reaction, where a molecule of water is produced as a byproduct. As you can see to the left of my head is the H2O molecule. And you can see that if you have two alpha glucose monosaccharides, they produce maltose, which is a disaccharide. So the breaking of the glycosidic bonds is called hydrolysis. So let's just remind ourselves of that. So here we go. So you can see condensation join monosaccharides together, whereas hydrolysis reactions break polymers apart. Glycosidic bonds form as water is released, whereas in hydrolysis that breaks the bond using a water molecule. And in condensation a disaccharide is formed, whereas in hydrolysis a monosaccharide is formed. So let's have a look at an animation of how a disaccharide is formed. So there are three common disaccharides you'll need to know. You need to know maltose, which is two monomers of glucose, alpha glucose, sucrose, which is made of glucose and fructose, and lactose, which is made of glucose and galactose. As you can see, some of the roles there. Maltose, there are other roles in animals related to their function. And lactose is a sugar found in milk, and it's an energy source for suckling mammals. Right, so obviously disaccharides join together and they then make polysaccharides, and they're made of many sugar molecules joined together by glycosidic bonds. So sugars join together through a condensation reaction. There are three main examples that we're going to need to know, which is going to be starch in plants, cellulose in plants, and glycogen in animals. So first of all, starch. I'll move out of the way. There we go. So starch is a storage carbohydrate. It's found in plant cells. It's made from two types of polymers, which is amylose and amylopectin. Both polymers are made from alpha glucose. So what's the function of starch in plants? Well, it's the main storage carbohydrate. Also, it stores excess glucose as it's too large to leave the cells, and it is insoluble, so it doesn't dissolve in water. So this means that it doesn't affect the water potential of the cell. Starch can be also hydrolyzed, obviously, to release the glucose for respiration. Now, looking at its structure, it's made up of two main molecules, amylose. So it's alpha glucose, and it's linked by alpha 1 to 4 glycosidic bonds. And this coils, as you can see in the picture, makes it compact, and therefore it stores more starch in a small space. And then amylopectin has a branched structure, so the side chains bond to one another via alpha 1 to 6 glycosidic bonds. So this means it's branched out. This increases surface area, allowing enzymes to hydrolyze those glycosidic bonds and therefore releasing glucose very quickly. You might get exam questions where they talk about, well, what are the key structures and how does that affect the function? So these are actual answers from AQA exam papers. So first of all, starch is a helical or spiral shape, so it's very compact to store more starch. They're large insoluble molecules, so osmotically inactive. Okay, so therefore it doesn't change the water potential. Branched, so glucose is easily released for respiration. Large molecules, so it cannot leave the cell or cross the cell surface membrane. Now, cellulose, again found in plants, in cell walls, it provides support and allows cells to become turgid. So they're mainly made, well, they are made of long unbranched straight chains of beta-glucose, and those have 1 to 4 glycosidic bonds. So almost making a grid-like structure. So very linear and then held together by hydrogen bonds, making them extremely strong. So key structures related to function: they're long, they're straight, they're unbranched chains of beta-glucose, they're joined by hydrogen bonds. So the actual strands are known as fibers or fibrils, so it forms micro and macro fibrils, and therefore it provides rigidity and strength to the cell wall. Glycogen is storage carbohydrate in mammals. It's found in large quantities in the liver and muscle cells. And glycogen is therefore an energy store, as it could be hydrolyzed to release glucose very quickly for respiration. Now, as you can see, it's very similar to what we've just been looking at with amylopectin. However, it has far more branching, and that's because it's made of alpha 1 to 4 and 1 to 6 glycosidic bonds. So key structures related to function. So it's obviously branched, so it can also store many glucoses. So it is actually coiled as well as branched. Polymer of glucose is easily hydrolyzed. It's branched, so that therefore the more ends for faster hydrolysis by enzymes. Glucose is stored, so it provides respiratory substrates for energy release. It's also insoluble, so it's not easily lost from the cell, and it doesn't affect water potential or osmosis. Okay, this is quite a useful little summary. This is from BioNinja, very good website, BioNinja, if you want to look it up. And it summarizes all of the features, the structures of all 3 polysaccharides. And this is just a quick kind of looking at the differences. So similarities and differences. In exam questions, they love you to talk about similarities and differences, and usually in the same line. So here we've got the similarities and differences between cellulose and starch or glycogen. So if we look at the similarities, both made of monomer glucose, they're all made of glucose. However, cellulose is made of beta glucose, whereas starch or glycogen are made of alpha glucose. They're both formed by condensation reactions, but cellulose is made up of straight, unbranched chains, whereas starch and glycogen are coiled or branched structures. Both contain glycosidic bonds. Cellulose, both are insoluble. Cellulose provides strength, and starch or glycogen have a storage role. Okay, I hope you found that useful. That is part 2. Part 3 is, well, actually it's part 2b actually, which is the Benedict's test. So how do you test for sugars, for reducing and non-reducing sugars? So please check that out. And part 3 coming up soon, which is proteins. So I'll see you soon.